In this video we will discuss about the paper that talks about this glycocalyx can be debulked by cathepsin protease. So for those who don't understand what this glycocalyx is the we can say that physical barrier around the cells, and this glycocalyx mainly composed of carbohydrate structures. So in the case of the cells which are not healthy or the cancerous cells, this glycocalyx thickness can be increased significantly, that it can act as a physical barrier that helps in the immune evasion. That is, it prevents the immune cells to come and detect the cancer antigens that are expressed on the tumor cancer cells. So in order to make the immune system sensitive, or again make the immune system efficient against the cancer cells, it is essential to remove this glycocalyx or this debulk this glycocalyx. So behind that idea, the paper talks about this, how this glycocalyx can be debulked. For that, they are able to find out that this cathepsin protease seems to be a probable candidate for debulking this glycocalyx. First figure talks about the in vitro studies, where what they did is they use the cathepsin to degrade MUC16 and try to find out the optimum conditions at which this MUC16 can be degraded. And why this MUC16? Because MUC16 is one of the abundant found carbohydrate structures in the glycocalyx in most of the cancers. So in the in vitro studies, they use this ovarian cancer cell 3 cells, and in that they took out the supernatant and did the size exclusion chromatography to enrich this MUC16. So once they get this purified MUC16, then they label them using this IR dye 800 CW NHS ester reactions through the NHS ester reactions. And thereafter, once they have this IR 800 MUC16, then they use different cathepsin for 16 hours at 37 degree, and they vary the pH. Basically they use 3 different pH, and once the digestion is done, then they use this in-gel fluorescence using this SDH page, then they use this Western blot using this SDH page, and then measure the protease activity using this in-gel fluorescence. So this is their experimental plan, and then in the results what they are able to find out that, and since this MUC16 has a high molecular weight, so in the case where protease didn't work, they can only see the signals that are at the top, and where the protease works, they are able to see the fragments in the lower molecular weight, or the smear of fragment shows the higher protease activity. So we can see that, and the potential cathepsins that works at different pH are highlighted in this pink color, and these preliminary results are based on their in-gel fluorescence indications in this SDS page experiment. And we can see that the potential candidate at pH 5 is cathepsin D, E, F, G, K, L, S, W, S, V, W, and these are the control STC, E, ESTCE. And similarly for the case at pH 6, different protease found to be more efficient is protease GKSB, not different, less protease found to be more efficient. And similarly for this pH 7, what they are able to see, the protease GKS are found to be more efficient. So as what the trends that we are able to see is that as they increase the pH, the protease activity seems to be goes down, indicating that at low pH or in the acidic conditions, the cathepsin protease can be able to cleave this MUC16, and it could be the potential candidate to study further about its activity to cleave this MUC16 in the actual TME environment or in the actual biological conditions. So in the figure A, they only shows their in vitro results we will discuss in our subsequent videos what are the other results that they have shown in the main figure, and what are the final conclusions, or how they come up to these conclusions that glycocalyx can be debulked by this cathepsin protease. That's all for this video. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.